Hi all, Dr. Clark here again, and today we're going to finish up some evolutionary information with evolution in action. I'm going to give you some examples of some studies where researchers have shown organisms to have evolved within a given amount of time. And so there's some classic examples with, say, guppies and Vistula moths, or what we call salt and pepper moth, or plethodon salamanders. There's quite a few examples of situations where evolution has been documented fairly readily in organisms that typically reproduce quickly. Okay, we can show evolution very quickly in bacteria and some other organisms because they reproduce so fast, but uh, the difficulty comes in organisms that reproduce slowly, because there's not enough potential change in the population's genes over time to really document evolution in action for a lot of those individuals. But nonetheless, I'm going to go through a few examples and show you, ways at which organisms have evolved due to different situations. Okay, so the first one that we're going to look at is a plethodon salamander example, and I'm going to have you watch a little video on the plethodon salamanders of California.
Meet a type of terrestrial salamander found in California. Now meet another, and another. These salamanders are members of the same species, so why do they look and behave so differently? In California, populations of these salamanders became separated geographically, evolving over millions of years to adapt to different environments. They've changed so much that they are on their way to becoming entirely separate species. David Wake of the University of California, Berkeley explains.
The history of this species complex began about 10 million years ago in the ancient redwood forests of northern California, where we have today this form picta, which is highly variable both genetically and in terms of coloration. There were two migratory routes followed by animals as they moved into southern California. One along the Sierra Nevada mountain chain, where animals moved into the forested region, the other along the coastal mountains. They avoided the Great Central Valley.
The populations that gradually moved down the forested region of California relied on blending in with their environment. Animals with good camouflage tended to survive more and produce offspring with similar traits. As the salamanders progressed south over millions of years, the markings became more distinct.
At the end of the chain in Southern California, we have this form Clauberi, which is the extreme of variation, showing the largest blotches, the most bold marking. These were all then camouflaged organisms that were hiding from predators. Along the coast, a different strategy was followed. Here, the organisms adopted the color pattern and behavior of dangerously poisonous newts, and they became, instead of camouflage, they were advertising their resemblance to these dangerously poisonous animals and gaining an advantage because of their mimicry. So what happened is that you had two differently adapted lineages moving to the south, and by the time they reached the southern end, they had essentially evolved into different species. But at certain areas, for example, in the Palomar Mountain area in San Diego County, we get these animals which are hybrids, and the hybrids are neither similar to the dangerously poisonous animals, they're not good mimics, nor are they good animals in terms of camouflage. So these organisms are essentially dead ends.
These hybrid offspring are not well adapted to their environment and are therefore less likely to survive. The two parent subspecies that produce the hybrids are well on their way to becoming entirely separate species, evolving just as Darwin described in his theory of evolution.
I think Darwin would love this example because it shows exactly the sorts of of patterns that he was talking about. Gradual adaptive divergence, leading eventually to the establishment of new species.
Okay, so there you can see an example of plethodon salamanders in California. One version is camouflaged with splotches and matching their background, and then one is highly visible and basically showing that they're poisonous, even though they're really not poisonous. Now, newts are poisonous, fire-bellied newts and things like that, that occur in the same region, and they're probably mimicking those color ranges, so predators just assume that they're poisonous. All right, so we already talked about sickle cell anemia and the facts that humans that have sickle cell normally occur in regions that have a high case of malaria, and that's because individuals that have sickle cell, or at least some of their cells are sickle shaped, will reject malaria. Okay, so they're immune to malaria. Malaria can't penetrate the sickle cell red blood cells. Okay, and so it might be an advantage for humans in regions where malaria is high to develop sickle cell. You might survive better than individuals that do not have sickle cell. Okay? And you can see an example of that from a human standpoint. Okay? One of the most classic examples of evolution is the peppered moth, or Biston bidilaria. Okay? And this research was done in Europe. These moths come in two main varieties. Either they're kind of a white with little black specks, or they're black with kind of brown specks. And when we examined peppered moths prior to the mid-19th century, almost all the individuals that you would see would be white or light in color. Okay? And then in the mid-19th century, mid-1800s, so about 1850, we had a big industrialized boom, and we had what we call the industrialized revolution. And to say the least, there was lots of factories, and they were burning massive amounts of fossil fuels, and there was no check on air pollution whatsoever. And so after the 1850s, okay, you really started to not see white or light colored moths any longer, and you saw the dark colored moth appear much more frequent. So, researchers were interested in this time period. Well, could Darwin's theory of evolution, could it explain these differences in color patterns in the peppered moth? Okay. Well, enough research has been done now, and was done at the time, to suggest that this is what we call industrial melanism. Right? That organisms that occur in regions where the industrial revolution started really will start to develop patterns or characteristics, plumage, color patterns that fit the soot or the dark colors that were coming out of the Industrial Revolution. Because dark organisms can better conceal themselves on darkened, soot-ridden buildings and trees and bushes and things like that, you start to see dark organisms show up more often. It wasn't just moths, it was lizards and all kinds of things. You start to see them show up more often. The dark variety, or the darker variety, survived, and the lighter variety was consumed by predators. So J.W. Tutt first hypothesized that this was going on, industrial melanism was going on in the 1890s, but it really wasn't until the 1950s when Bernard Kettlewell came up with an experiment to test industrial melanism. And Kettlewell's experiment was fairly simplistic. He would catch white moths and he would catch black moths, and then he would pin them or glue them to different colored backgrounds, trees that either he had scrubbed with a scrub brush to remove the soot and show their white bark, or he would leave them, with the soot on them and used, dark moths and white moths on a dark background, or dark moths and white moths on a light background. And he'd just glue them to the tree, and then he'd sit back with a pair of binoculars and watch the predators. Okay? And sure enough, what he found was if you're a light moth on a light background, you got consumed or eaten far less than if you were a dark moth on that white background. And vice versa. If you're a white moth on the dark sooted background, okay, you got ate far less than the dark moth on the dark background. Okay? And what we see from the research is that when we start looking at air pollution and we start looking at, a period of, the 1900s, 1959, all the way really to, present day, you start to see this drastic change back. So this is the percent of moths that are melanic, okay, or in other words, the percent of moths that were black. Okay? In the 50s, the 1950s, they were all black, over 100%. Okay? And the way you can get over 100% is based on some logistics with the study in which he was examining, trees that had natural moths also. So he was putting moths on there artificially, but then there was actually natural moths. So his populations were bigger than his percentages. But nonetheless, in 1956, in England, they had a clean air legislation, so like a Clean Air Act, just like the United States did in the 60s. But in 1956 they did, and you start to see this change that after the amount of carbon dioxide and carbon fiber and the amount of smoke and soot was decreased from plants, the amount of air pollution decreased, so did the amount of black moths. So it took a little bit, it took a lag period, quite a bit, but eventually, in the time we hit, the 1980s, you start to see a drastic change all the way to today, where about 10% of the moths are dark and 90% are back to white. So this is, that shift in the population's genetics, shifting from white to black, during the Industrial Revolution, and then after the Industrial Revolution, when the Clean Air legislation and act came about, going back from black to white, okay, and you see these patterns. It's not that the white moth was ever gone or the black variety ever left. Okay? It's just they occurred in very small populations because birds were picking them off of the habitat. Okay? And so, like I said before, the results clearly show that the frequency or the number, the percent of dark or melanic Biston botuliuria, decreased as you get decreases in air pollution. Okay, so how about a guppy experiment? Most of you guys probably know something about guppies or seen guppies. Guppies are really colorful aquarium fish. They are native to Trinidad. And the reason why a lot of people like guppies is they give live birth. So it's really easy to buy a few guppies, a male and female, a couple males, a couple females, put them in a tank, and then next thing you know, if the conditions are right, you have hundreds of guppies. Okay? And they're colorful. Okay? But they're native to Trinidad, and they're found in a couple different types of environments. but two environments that were important for researchers like David Resnick and then David Resnick's grad students, which we'll talk about some of their research here. But their idea was these guppies are found in a couple different environments. Okay? One environment where there's these pools, okay, and these waterfalls are filling these pools, and these pools have guppies and then another species of fish which we call a killifish, and the killifish sometimes consumes guppies, but it's not a high on the list of predators to guppies. Okay, so rarely do the killifish actually eat the guppies. Okay, so you have this pool of water, native habitat, where the guppies are safe from predators within inside the water. Okay? And then you have these pools below the waterfalls, okay, where the guppies are found with a cichlid predator, which is called the pike cichlid, and this is a piscivore, so it eats other fish. It's a predator of guppies. Okay? So the classic experiment was, as long as there's a little bit of gene flow, okay, guppies will be different in the pool with no predators than in the pool with predators. Okay? So this is kind of the classic setup of an example. Now, there is a little bit of gene flow, so you can see some of the traits come back, especially with high waters or floods and things like that. But for the most part, if this is done in a small amount of time, you can get drastic changes in the population. Okay? So what are the differences? Well, when we look at the guppies that are below the waterfall, where they're encountering these cichlid predators, okay, the pike cichlid, these guys are not very colorful. Most people wouldn't even be able to tell that they're actually guppies. Okay, so not very colorful. They tend to reproduce at a much younger age. Okay? They don't get as big. There's quite a few differences. Okay? Okay, so again, no color, very little color, reproduces small age, okay, or early in life, and a small size. Okay? So researchers suggested, well, this is natural selection, that it's the predators that are selecting against color. Okay? And the predators are driving guppies to reproduce sooner and at a smaller age, or a smaller size. So younger age, smaller size. Okay, so this is kind of what the system looks like. Okay, you would have a waterfall that's flowing into this pool, and you have some guppies that got these colorful patterns, which we often call eye spots on fish. You have this killifish that occurs in here. Sometimes eats guppies, but not very often. And then you have this other pool where guppies occur, and they don't really have much color. You can't really tell males from females. And you have this giant piscivore fish, the pike cichlid, which is going to eat as many guppies as it can. And that's really the system. Okay. So John Endler was a grad student of a guy named David Resnick, and co-authored a bunch of papers on this system. They conducted experiments that really were to determine whether or not predation, or the risk of predation, was enough to drive evolution. So what did they do? They went to Walmart and they bought a bunch of kiddie pools, because that's what fish scientists do. You go to Walmart, you buy kiddie pools, and then you set up what we call mesocosms. So these small habitats in which you can manipulate just a couple traits and see what occurs. So what they did is they set up a few different systems, a few different conditions. One where there was no predator present, one where there is a killifish present, so low predation risk, and one where there's that cichlid present, and that's high predation risk. They took guppies and they placed them in these mesocosms into these laboratory settings, okay? And then they saw what would occur. And really, after about 10 guppy generations, okay, the individuals in the low predation risk were much more colorful than the individuals in the high predation risk. And so no predators were the most colorful, but so most colorful, still colorful, not much color at all. 10 guppy generations is about 10 months. A guppy will reproduce about every 20 days. Okay, so in less than a year, they could drive a population to be drastically different, genetically speaking and phenotypically speaking, when you look at the colors. Okay? So then he redid the experiment. They redid the experiment in Trinidad in the streams by blocking the streams off and pulling all the fish out and redoing it just to make sure that it wasn't just a artificial example or laboratory situation that was causing these drastic changes, but they see the exact same results in the field. And this is what you see. You see those colorful individuals with bright spots, etc. Where there is no predation or low predation risk, and where there's high predation risk, it just drastically decreases, and you get these female lookalikes. Okay? And so you can see that over a very short period of time, you can get evolutionary change in the population where there are no brightly colored individuals left in high predation populations. Okay, so next time we're going to talk about speciation. We're going to talk about what determines what a species is. So I gave you an example of those plethodon salamanders, going down the Sierra Nevadas, going down the coast mountains, okay, and that they're all the same species right now, but they're on their way to becoming separate species. So what does it mean to be a species? What does it take to be a separate species? And we'll look at that when we come back next time.